I will be presenting our study, which is titled Untargeted Metabolomic Identification of Diagnostic Biomarkers in Ectopic Pregnancy. The annual incidence of ectopic pregnancy has increased over the past 30 years, becoming 20 cases per 1,000 pregnancies. It is the cause of 2.7% of all pregnancy-related deaths. Currently, the diagnosis of ectopic pregnancy is based on a combination of serial quantitative HCG testing and transvaginal ultrasound. Although these tools have enhanced our ability to detect the disease, repeated evaluations are often required and early diagnosis still remains a challenge. Pathologies such as pertubal cyst, corpus luteum, hydrosalpinx, endometrioma, or bowel could easily be mistaken on transvaginal ultrasound for an ectopic pregnancy. The most recent ACOG practice bulletin that came out in 2018 has also made a remarkable change in discriminatory HCG level. It is recommended that the discriminatory value should conservatively be high, which is 3,500, to avoid any potential misdiagnoses. Therefore, there is a larger patient population with pregnancy of unknown location that requires a serial follow-up throughout ectopic pregnancy. The rapid development of systems biology and the omics cascade, including genomics, transcriptomics, proteomics, has forged a new era of research in understanding systems biology. Metabolomics is the newest member of this family. The highly dynamic nature of the metabolites makes metabolomics the endpoint of omics cascade, which yields the closest profile to physiological phenotype. It is the field of life science research that uses high-throughput technologies to identify and characterize all the small molecules. In other words, metabolomics reflects the state of cell, organ, or organism. This slide presents metabolomic platforms. Due to vast chemical and physical diversity of metabolites, there is no single analytical method that can currently measure the concentration of all metabolites. There are two different approaches to obtain metabolomic profiling, targeted and untargeted. Targeted is an approach that explores previously identified molecules. However, it only looks for a small number of metabolites. It is hypothesis testing. In other words, looks into certain metabolites pathways that are planned. In untargeted approach, however, we globally look at the full spectra, almost includes more than 10,000 molecules. However, the difficulty is in the identification of metabolites. Hence, it is hypothesis generating, which explores the unknowns and develops new biomarkers. In 2019, in order to test our hypothesis of ectopic pregnancy having a different metabolomic profile, we published a study focusing on targeted metabolomics using NMR platform. Our study was able to identify significant metabolite markers with high prediction rates. Biochemical pathways revealed markers related to early onset inflammation and altered trophoblast invasion prior to clinical evidence of an ectopic pregnancy. Hence, following our previous study, we had two main objectives overall. First, we aimed to identify potential metabolite biomarkers by using untargeted metabolomics approach. Second, our aim was to elucidate alterations of biochemical pathways that are associated with ectopic pregnancy. This slide presents our recruitment flow. In order to ensure the diagnosis of ectopic pregnancy, only the cases that underwent surgical treatment included in the study, and the tubal pregnancy was histopathologically confirmed with the presence of a chorionic villi at the salpingectomy specimen. Cases were enrolled prior to methotrexate or surgical treatment. Patients that were hemodynamically unstable or with ruptured ectopic pregnancy were excluded from the study. We performed tandem mass spectrometry analysis on plasma samples. In total, 50 ectopic pregnancy cases and 50 age-matched intrauterine pregnancy controls were included. In order to identify metabolites, we used GMPS molecular networking, which matches our data with already published 1,000 metabolite libraries, which I will mention in the results section. To avoid any cross-fitting, our sample set was separated into two groups, discovery and test group. Discovery group was used to develop our diagnostic models, and it was validated on a test group. Univariate and multivariate analysis were performed to see the separation of metabolomic profile and to develop logistic regression models. There were no significant difference in demographics between groups except prior ectopic pregnancy and anaxial surgery. Transvaginal ultrasound showed an axial mass finding at 67% of all cases. In total, we visualized 585 molecules in our spectra, and 46 of those were significantly different in ectopic pregnancy cases. As shown at the first plot on the top left, metabolomic profile showed significant separation between cases and controls. Our top 2 metabolite model was discovered and tested with a high accuracy. It achieved an area under curve of 0.962 with 100% sensitivity and a 95% of specificity. As also seen here at the top right, multiple models were developed and also showed with high area under curves, which are coded with different colors. At last, to identify metabolites and identify altered metabolomic pathways, we performed molecular networking. The picture on the right shows the full molecular network that was obtained for ectopic pregnancy. We were able to focus on a certain group of metabolites and able to see which are altered in ectopic pregnancy. And we were able to identify activated glycerol phosphocholine pathway and significantly depleted levels of sphingolipids in ectopic pregnancy. So just to comment on these findings, we report the first time untargeted metabolomic profiling of ectopic pregnancy that appeared to be highly accurate for detecting the disease. Metabolomic analysis also provided new insights into the dysregulated metabolisms associated with ectopic pregnancy, which was supported in the literature that these pathways are related to fetal-maternal interface and also cell membrane invasion that is disrupted by trophoblasts. As a conclusion, metabolomic biomarkers could potentially facilitate the early diagnosis of ectopic pregnancy and reduce the mortality and morbidity. Despite these promising results, of course, there are several limitations of our study. One of them is that we did not include spontaneous or threatened abortions, which can be differential for an ectopic pregnancy. And also, the population studied here was ethnically homogeneous. Larger studies, including other non-viable pregnancies, are warranted for the future. Thank you.